Good morning. Just making sure everything's working. Do seem to have volume. Camera seems to be going. All right. So let's start with the cytoskeleton. This will be a wrap up of chapter 3. I hope to finish it today. Well, I'm going to finish it today, but I hope to finish it in this one recording session. With the cytoskeleton, we talked about all the rest of the organelles of the cell. So let's talk about the cytoskeleton. The cytoskeleton is a dynamic structure which changes the function based on the needs of the cell. It gives the cell its characteristic shape. So if a shape, later when we start talking about squamous cells and cuboidal cells, it's the cytoskeletal elements that gives it that shape. Also provides strength, structural integrity. Cytoskeletal elements allow for cellular movement. We're going to talk about the flagella and the cilia in a moment. And perform specialized functions in different cells. For example, in the phagocytic cells, it allows that cell to move, but also to engulf those things that it's trying to eliminate, and also contraction of the skeletal muscles and the different muscle cells. There are 3 types of filaments found in the typical cytoskeletal system. The actin filaments, these used to be called the microfilaments, and some textbooks still refer to them as microfilaments. We call them actin filaments because the product they are made up of is a molecule called actin. There are intermediate filaments, excuse me, and then there are the microtubules. The actin filaments, again, also known as microfilaments, are the thinnest of the filaments. They are composed of 2 intertwined strands of actin subunits. Don't worry, I'm not going to ask you to remember that they're 5 to 9 nanometers thick, but these things provide structural support, they bear tension, and they help maintain cell shape. When we start talking later this week and next week about tissues, and we talk about those tissues that give cushioning and are weight-bearing things, have a lot of actin subunits. Also can be involved in cellular motion when involved with a motor product called myosin. We're going to find out later on toward the end of the semester when we start talking about the muscles. We're going to talk about lots of these fibers along with a protein called myosin that makes the muscles be able to do the things they do. The intermediate filaments are more of a rope-like structure. They're made of the protein keratin. These are found in things that are more permanent. The structures within the different cells, when we start looking at different types of fibers that are found in cells, those fibers that are fixed don't tend to change shape much. Those are primarily made up of the intermediate filaments. These help organelles and nucleus maintain both shape and size. Again, keyword here, maintain. And they help cells and tissues withstand mechanical stress. Again, those things that have to do a lot of flexing. Cartilages and ligaments will have lots of intermediate fibers. The microtubules are the largest of the filaments. They are also, key characteristic here, they are hollow, composed of a product called tubulin. They can be either rapidly added or removed, allowing for the size and shape of within the cell. So those cells that are available to change shape, or available, that have the ability, is a better way to put that, to change shape, probably have lots of microtubules in them. Certain motor proteins like dynein and kinesin allow for transport vesicles and microtubule network, and we'll talk more about those transporting things later on. So the different types of actin filaments you see here in these nerve cells, I believe those are nerve cells. Then we have intermediate filaments found here, and the microtubules were found in this particular type of cell. You see they've got them stained in a very characteristic color, so you can see here's the microfilament here. This whole structure is primarily made up of mostly these, and then this structure is made up primarily of those. Microtubules extend out from a centrosome. When the cell is not dividing, the centrosome is a microtubule organization center located close to the nucleus. A pair of centrioles is critical for cell division. In a couple of chapters, when we start talking about metaphase and anaphase and all those kinds of things, we're going to talk about the role of the centrioles. And then the basal bodies are modified microtubules where the flagella and the cilia originate. We'll talk about cilia and flagella in just a moment. But there are the centrioles, and you see they've got them on this transmission electron microscope, magnified 137,000 times. So a very large magnification. And you see then the microtubules will hook and start to grow outward. And when we start talking about spindle fibers forming in metaphase, we will talk about these structures. Cellular extensions, we have three different types. We have the microvilli, we have the cilia, and the flagella. The microvilli are finger-like extensions of the plasma membrane. They are made primarily of actin. They help maintain shape. And this is that whole structure and function core principle. Cells are designed to do the job they do, and they can do the job they do because of the way they are designed. These increase the surface area of the cells for absorption. For instance, these types of structures are going to be found lining your small intestine. Greatly increases the surface area for nutrients to be absorbed. I mean, think about it. If this was just a flat surface, you would have that one little area there, however big, length times width of that would be. But by adding all of these little microvilli, you get all this increased surface area to absorb all those nutrients that are moving through the digestive system. The cilia are a hair-like extension projections. Also, they are microtubules and motor proteins. They move in unison to propel substances past the cell. Now remember, when we were talking earlier, I may have mentioned protist organisms and the one-celled organisms. Some of those, the cell itself moves through cilia, but for us, the human organism, our ciliated cells, our ciliated cells are designed to move this way, and they're going to come back up the opposite way. And ultimately what that does is it creates a current, so things are moving this way past the cell. And these are found in great numbers where they do exist. The flagella is a solitary structure, longer than cilia. In the human organism, it is found only in sperm cells. So theoretically, the only flagellated human cell is the sperm cell, and then theoretically, only half of the human beings make those. But as we all know, there are not half as many males as there are females. There are more females on the planet than there are males. The flagella and cilia are structurally similar to the centrioles, except they contain two central microtubules not found in the centrioles. And again, here's the sperm, and what it does with this long flagella is it creates a whip-like structure or whip-like motion, and that pushes the cell forward. And then there are the different versions. Here's the microvilli as it appears. This is a blown up sort of magnified showing the cilium there. And then there's the different structures. So we have three different types of cellular extensions again. We have the cilia and the flagella, and then the microvilli, which this particular table does not show. The cilia there, again, very, very numerous. Flagella on the sperm cell, it's always singular. There is a disorder called primary ciliary dyskinesia, which is a genetic disorder. It is a defect in one or more protein components of cilia and flagella. This affects cells of the respiratory passage primarily, but can also affect the inner ear, uterine tubes in females, and sperms in males. The major side effect of this disease is a buildup of mucus in the lungs. With that buildup of mucus, in addition to an increased labored breathing. You can also have an increased risk of infection. You can then have repeated damage due to infections and mucus plugs which form in the lungs. Repeated ear infections may lead to hearing loss, and the males who suffer from this tend to be infertile because their sperm are not motile because the flagella is malformed. The flagella doesn't work properly, then it can't swim and get to where it needs to be. The nucleus of the cell. The nucleus is the governing body. It's the central command. Largely determines the types of proteins and the production rate of those proteins. In humans, our DNA is found in the nucleus. The DNA is the code or plans for nearly every protein in the body. There are some few proteins that can be made outside of nuclear control, but for the most part, if it's a protein, its reproduction and synthesis is driven by the DNA found in the nucleus. These plans or genes within the DNA are executed by several different types of RNA to build a wide variety of proteins. And here in just a moment, we're going to talk about several different types of RNA, and it would be very informative for you on your first exam, which is coming up really shortly. There will be an essay question about protein synthesis. The nuclear envelope is the membrane that surrounds the fluid or the nucleoplasm. There are lots of components found within the nucleoplasm, but primarily water, free nucleotides to make more DNA and RNA, the enzymes to assemble those, and then other proteins. Then there is the DNA and the RNA. DNA and its associated proteins in the nucleoplasm are what is referred to as a loose structural arrangement called chromatin. Some folks may pronounce that chromatin. Again, I was taught chromatin. I've heard it pronounced chromatin in the past. And then one or more nucleoli are suspended within the nucleoplasm, and we'll talk about the nucleoli here in a few minutes. There's the nucleolus right there, which is the singular term of nucleoli, which I just mentioned. Here are the nuclear pores. Nuclear pores are where the messenger RNA and other things that need to exit the nucleus are going to leave. The nuclear envelope, again, is the membrane. There's the rough endoplasmic reticulum. Nucleoplasm is found here, the little heavily looking substance. The nuclear envelope, again, double phospholipid bilayer. Virtually every membrane in the cell is a phospholipid bilayer. Similar to that of the mitochondria, there is an outer membrane, which is studded with ribosomes. There is an inner membrane, which lies interior of the nucleus, supports the network of intermediate filaments. And then there are the nuclear pores, which, again, they're large proteins where the items that need to exit are going to leave. Blow up of the nuclear pore. There's the phospholipid bilayer right there. There's a protein holding this structural protein in place. There's the nuclear pore where things are going to exit. Here's the outer membrane, the inner membrane, and the nuclear envelope there. Chromatin is an extremely long DNA molecule and its associated proteins. It is designed to organize and fold the molecule to conserve space. The nucleosome is a strand of DNA coiled around a group of histone proteins. So this is the nucleosome surrounding the histone protein. By doing this, you reduce the length of this strand by approximately a third. So you can put 3 times as much length in a given space by the cell wrapping the DNA. During cell division, the chromatin threads even more tightly, or even coils even more tightly, and condenses into that structure that we refer to as a chromosome. Human cells contain 2 sets of 23 chromosomes, one from your mom, one from your dad, so one maternal, one paternal, for a set of 46. Sister chromatids, which we will talk about, are identical copies of each chromosome, made in preparation for the cell division. connected to one another at the centromere. So don't get confused when you see centromere and centrosome that we talked about a few minutes ago. So this is the completely relaxed stage of DNA. Then the histone proteins come in, form the nucleosomes. Those begin to coil even tighter and tighter and tighter until you wind up with the entire length of the DNA in this very short, compact structure called a chromosome. This is a human karyotype showing the different chromosomes. Here are the two sex chromosomes, the X and the Y. The really large one is the X, the much smaller one is the Y. And the nucleoli is the nuclear region responsible for synthesis of ribosomal RNA, or rRNA, and the assembly of ribosomes. Protein synthesis. This will be, starting here, this is a clue. This will be an essay test, an essay question on your first exam, a thorough discussion of protein synthesis. So, protein synthesis. Manufacturing proteins from DNA blueprint using RNA. What is gene expression? That is the production of a protein from a specific gene, which includes two processes. Transcription, which is where the gene for a specific protein is copied from the DNA. That copy that is made from the DNA is referred to as messenger RNA, or little mRNA, and this little mRNA exits through the nuclear board into the cytosol of the cell. Translation occurs in the cytosol. The messenger RNA is going to bind to a ribosome. That initiates the synthesis of a polypeptide chain, which is where a specific sequence of amino acids will be added to that growing chain based on the codes found in the messenger RNA. So DNA, through a process called transcription, will make messenger RNA. That messenger RNA then will be translated by the ribosomes, and we will result in a protein. What is a gene? Everybody hears about genes. I have brown hair. When I had hair, I had brown hair because of my genes. I have green eyes because of my genes. A gene is a long chain of nucleotides that determines the sequence of amino acids in a specific protein. There are 4 different nucleotides in DNA. They are adenine, thymine, guanine, and cytosine, abbreviated as A, T, G, and C. We've already talked about those a little bit in an earlier chapter. Each set of 3 nucleotides, or each triplet, represents a different amino acid, and each amino acid may be represented by more than one triplet. Those triplets are referred to as a codon. So during transcription, each DNA triplet is transcribed into its complementary RNA copy, and each 3 nucleotide sequence of messenger RNA copy is referred to as a codon. During translation at the ribosome, each codon is paired with a complementary tRNA, and the T here stands for transfer. The tRNA triplet is referred to as the anticodon. It's the opposite of the codon, ergo the anticodon. And it has a specific amino acid attached to it. That amino acid will then be added to the growing peptide chain. And your genetic code is the list of which amino acid is specified by each DNA triplet. And there you go. There are the abbreviations for all the amino acids. There are all the different 3-letter codes you can make out of U and C and A and G. And remember, this is RNA, so it's going to have a U for uracil in place of the T for thymine. So as you can see, if we multiply these, there's 4 here, 4 here, 4 here, and 4 here, and then there's 4 rows. That's 64 different possible codons. And if you count these up, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, you've got 20 amino acids. So if you've got 20 amino acids and 64 codons, then clearly there are going to be some amino acids, as you can see, that have multiple codons. There's a couple of important ones. One especially, AUG, that is referred to as the start codon. It also just happens to be a codon that adds methionine to the chain. And then up here on the upper right, there are 3 stop codons, UAA, UAG, and UGA. So 3, University of Auburn, Alabama. The University of Alabama is really good. And the University of Georgia at Athens. Those are your 3 stop codons. Whereas school starts in August, the 3-letter abbreviation for August is AUG, and for AUG is the start codon. Mutations are changes in the DNA due to a mistake in copying the DNA or induced by agents. Those agents are called mutanogens, and we will talk more about mutanogens later in the course. Common mutanogens are ultraviolet light. Well, where do we find ultraviolet light? I'm standing under it right now. The light emitted by these tubes is ultraviolet light. Some other forms of radiation, chemicals, are very common. Used to be a very common chemical used in industry, not so much anymore, is benzene. And the reason it's not used is we have discovered that it is a mutanogen. And infection with certain viruses, and we'll talk about viral infections later on in either AMP1 or AMP2 that cause certain mutanogens or cause certain mutations. And then DNA mutations are the basis for many diseases such as cancer. The death cap mushroom, there's the scientific name of the death cap mushroom, Amanita phalloides, responsible for 95% of the mushroom related fatalities worldwide. Why is that? Because, well, it's tasty, for one thing, even though it's incredibly toxic. It resembles many non-toxic mushrooms. And what does the toxin do? It inhibits RNA polymerase, which prevents the formation of new strands of RNA. It basically stops you from making proteins, and that is not good. Stops protein synthesis, disrupts cell functions, and leads to cell death. Currently, there is no antidote available to this particular toxin. Some early stuff is beginning to show promise. Those people who do survive this usually require a liver transplant due to liver damage. That is one of the reasons you don't go out into the wild and pick your own mushrooms for your salad and your sauteed mushrooms and all of that kind of stuff, because unless you are a mushroom expert, you may be picking one that you think is perfectly safe, but because this one looks so much like the safe ones, you may be picking one of the bad ones. One of my favorite TV shows is a British crime drama called Midsummer Murders, and they had an episode a few weeks ago on Arkansas Educational Television on Channel 2, and one of the victims of the murders was actually killed by death cap mushrooms. Transcription is the process of making messenger RNA. You take the product is called a transcript. It exits through the nuclear pore into the cytoplasm to the ribosomes. The enzyme that builds the transcript is called RNA polymerase. The RNA polymerase binds to the gene, brings in the complementary nucleotides, links them to form the messenger RNA. This is a process that has 3 stages. There is the initiation stage. That's where the process gets started. There's the elongation phase. That's where all the different I'm gonna speak the word. I'm gonna spit the word out here in a minute. The amino acids are added, or all the groupings are added, and then the termination phase is where the process ends. So initiation, the beginning of transcription. Protein transcription factors bind to the promoter region near the gene on the template strand of the DNA. So here's my template strand. You see it's going to open up. RNA polymerase also binds to the promoter, and DNA unwinds with the aid of DNA polymerase. So you have the transcription factors and the promoter attached here. They start to work. The two DNA strands open, and the RNA polymerase starts to do its job. And here is the new transcript being formed on this side of the DNA. Elongation is where the RNA polymerase covalently bonds complementary nucleotides to the growing chain of messenger RNA. So if there's an A here, there's a U here. If there's a C here, there's a G there. If there's a T there, there's an A there. Puts the complementary bases onto the chain. And here you see a blow up, so you can see those letters a little better. And then finally, the termination is when the last triplet of the gene is reached. The newly formed pre-messenger RNA molecule is ready for modification. So here's my RNA transcript. Ultimately, what will happen is this promoter will finally reach one of those stop codons, UGA, UAA, or UAG, and termination will occur. Big picture of animation. I'm not going to run the animation. You can run it if you want to at your home, at your leisure. But basically, you have DNA through the process of transcription makes messenger RNA. The messenger RNA through the process of translation is going to make proteins, and we're probably going to go over the translation phase next. So I'm not going to do these reviews. You can do those again at your leisure. And as it clicks, it shows you the correct answer. I'll go back and show you. Start. There we go. So you got question number 1, the process through which messenger RNA is turned. What is it? What is it? What is it? It's transcription. Transcription begins when transcription factors bind to the promoter. The enzyme that elongates the messenger RNA transcript is RNA polymerase. And during transcription, free nucleotides from the nucleoplasm are hydrogen bonded to complementary nucleotides of DNA template. Right. Oh, one more. And I said I wasn't going to do these, but I did them anyway. Transcription ends, and the messenger RNA transcript is released during which stage? Which one? Termination. All right, so transcription. After transcription, the transcript, the pre-messenger RNA, isn't quite ready. It must first be modified in several ways. The non-coding sections of a gene do not specify amino acids. These are referred to as introns. The sections that do specify for amino acid sequence are called exons. So you have introns and exons. And here we see this is the pre-RNA. All the different introns and extrons, we'll go about transcription. Then you get all these little areas here. And the introns are going to be the yellow ones. The exons are going to be the red ones here. So in RNA processing, the introns in the pre-RNA are removed, the exons spliced together. So we're going to take these sections out, bring these sections together, and we have a nice little messenger RNA that's ready to go and be translated into a protein. So again, big picture, DNA makes through transcription makes a messenger RNA. The messenger RNA is then has the introns removed, translated into a protein. So translation occurs at the ribosome, where the nucleotide sequence of messenger RNA is translated into an amino acid sequence with transfer RNA. So here's the transfer RNA here. C, A's and C's and G's and U's. Kind of looks like a key. The way I always learned to keep it close. There's the amino acid. Here's the anticodon. Now the anticodon is going to attach, the amino acid attaches on this end of the key. The anticodon is where it's going to attach to the messenger RNA. The anticodon is on one end, again down here. It's a sequence of 3 nucleotides, which are the complements to the codons of the messenger RNA. Each ribosome has 3 binding sites for transfer RNA. The A site binds the incoming transfer RNA to an amino acid. The P site, the amino acid is removed from its transfer RNA, added to the growing peptide, and each transfer RNA then exits the ribosome from the E site exit, where it's free to pick up another amino acid. So where the amino acid is attached, amino A site, the amino acid, P site, the growing peptide, and the empty tRNA, the E site, you can think of it either as empty or the exit. Translation is organized into 3 stages. Again, you also have initiation. Here's my ribosome. Here's my 3 sites, the exit site, the P site, and the A site. As you can see, P site is attached, moves along. This one will move into the A site to be picked up by an additional amino acid. The glycine then exits. You see the methionine stays attached. We're picking up this one. We continue to do that. There the exit of the GGG. Where's the GGG? Here. And then we exit and bring in the next new release factor. That's the new one coming in there. All right. That wraps up chapter 3. I was able to do it in one sitting. If you have any questions, have those ready for me on Wednesday morning. Also, it may take a couple of hours for this video to fully load onto the server and then get uploaded into YouTube. I will get that done as quickly as possible today. And then we will start on chapter 4 on Wednesday. And with any luck, everybody cross your fingers for luck, or knock wood, whatever you do for luck, we will be able to get through chapter 4 on Wednesday and Friday, and that will get us back up to speed. If not, what I will more than likely do is Friday afternoon, do another one of these little video sessions to wrap up chapter 4. So a week from today, next Monday, we can actually start chapter 5 and be back on schedule. We'll also be putting in an announcement about your first exam. If it did not open today, it should open one day this week, and I will adjust those dates so you have a full 7 days to do that. So again, essay question, first exam, protein synthesis. Have a good day.